which is basically ulcers in your colon. It actually affects a lot of people, 1.6 million Americans. Definitely not as high as, say, like Alzheimer's or diabetes, but it is a substantial amount of people. And one thing to note is that it is chronic, and the diagnosis is actually very early. So the average age of diagnosis is 21 years of age, which is pretty young, especially because it's chronic. So you have it for the rest of your life. And on average, patients have 2 surgeries, sometimes very painful, like removing the entire colon. So it definitely affects the quality of life a lot. And here's some just more statistics. It's quite a complex disease, both Crohn's disease and ulcerative colitis. You can see here more than 100 genes contributing to the yeah, so that's all I wanted to show. So I wanted to outline basically the current treatments that we have. And so, like I said before, there's surgery and there's hospitalizations in unfortunate cases. But most of the treatments that we have for IBD today is in biologics, basically therapeutics that are based around DNA and proteins. And of that, of those biologics, most of them are what's called TNF alpha blockers. That stands for tumor necrosis factor alpha. It's a protein that produces inflammation. So a TNF alpha blocker will block that inflammation. And this is actually a well-used biologic for other diseases as well, such as rheumatoid arthritis, because that one is also has to do with inflammation. So here's a really important statistic that we have for current treatments, and that is that the patient response, there's up to 40% of Crohn's disease patients that do not respond clinically. So they just become refractory, and they lose, I guess you can say, the ability to respond to these biologics after 6 months. So this is well documented. This is actually a big problem because this is the main treatment that we have for IBD to reduce the inflammation, and then we have almost half of these patients not being able to respond. So in response to this, our collaborators in the School of Medicine under the Ghosh Lab, they have a new strategy that's based in artificial intelligence, or AI assisted target identification using Boolean networks. So I'll just say it before I go on to this explanation. I'm the chemistry guy, and they're the biology people and the people doing all the math. So I am not an expert on Boolean networks, but I do want to try to explain some of the work that they've been doing. So basically, the strategy is to identify druggable protein targets that are key factors in deciding between healthy and disease states of patients. So in this figure, as you can see here, we start with Crohn's disease data set, and so they start with a very large human patient data set of the mRNA in both normal and Crohn's disease patients, as well as ulcerative colitis, but I do not show this figure here. And then we have a gene cluster, as you can see here. And this cluster is just many genes that are clustered together that are similar genes. And we have relationships. Maybe this is better explained in this pathway here, where, for example, C1 being cluster 1 of a bunch of genes are very high gene expression level in a normal patient, but very low in a diseased patient. And so is cluster 2. And cluster 3 would be very high in a Crohn's disease patient. And so here are some of these invariant relationships that we have here between these clusters, and you can map out all of these clusters into, and once these clusters are then mapped out in a continuum state, then you can go into each cluster and identify key proteins, most likely right around the state between normal and Crohn's disease. And so the assumption is that someone who is diagnosed with Crohn's disease might not be the same as someone else who's diagnosed with Crohn's disease. It's very unique. And by continuum state, what we mean is that it's the pathway along the disease that person has the disease. Essentially, if someone is far along the pathway, then they're very far from the normal state in terms of their gene expression level, and they have possibly a lot more symptoms than someone who has not gone along that pathway a lot more. So hopefully that makes sense in terms of that. Sorry if that's a confusing explanation, but yeah, let me know if you guys have questions now. You can definitely interrupt. Okay, so I will go on now to try to explain some of these key protein factors. So here I have written out the continuum state, but in a slightly different format. These blue clusters being each cluster. And through the work that the Ghosh Lab has done with their collaborators, they identified a gene for protein PRKAB1 being in cluster 2 as a key protein target, which can basically, when activated, this protein can stop the disease from going on. And we even have evidence currently that it might be able to even reverse the disease back on this continuum state. So taking this druggable protein target is where a collaborator's work stops and my work starts. So I just wanted to give a background for this protein target that we have. So AMPK stands for AMP protein kinase. It has 3 subunits, the alpha, the beta, and the gamma. And so like I was saying before, the beta is of most concern for us, specifically the beta 1. And so this protein is the cell's energy sensor. It's actually involved in a lot of diseases, including diabetes, neurodegenerative diseases, and diseases such as these. So a good way to understand this protein, it is kind of like the battery indicator on your laptop. So it tells the cell how much energy the cell has. And it basically does this using the ratios of AMP and ATP and ADP and ATP. So it would say like a high charge, 100% charge is when there's a very high ATP to ADP ratio. Back to the subunits, which are very important for my study. We have the two different isoforms for the beta subunit. So we have beta 1 and beta 2. And so going back to the protein target that's of interest, it's specifically beta 1, not beta 2 actually. And you can just see here in this figure real quick that the beta 1 and beta 2 have different expressions depending on the organism, mouse or human, depending on the tissue, whether it's skeletal tissue and whatnot. So this figure describes well kind of the workflow of our work collaborating with the Ghosh Lab's work. So like I tried to explain before, there was target identification using the Boolean networks to analyze the gene clusters and the disease continuum states. Then there are the drug targets that came about of this. 2 specific drug targets. I won't be talking about the PPAR alpha gamma targets today, but that is also a project that I'm also working on. And both of these targets are inflammatory bowel disease targets. Then onto the work that I and another member of our lab worked on is in silico docking, which I'll explain a little bit later. So molecular modeling using docking in the computer to design beta-1 selective agonists. And medicinal chemistry, which is the hands-on using of chemistry to make these molecules that you simulate in the computer. So just specifically designing and synthesizing these compounds to target the beta-1 protein. Then using the in vitro protein assays to test them. So kind of like, as we know, like a protein test before you get into the much deeper preclinical validation using mouse models. And there we're going a little bit deeper with targets that we think are good hits and good prospects for an actual therapeutic. And the blue here is the Ghost lab and what they'll be working on. So once these molecules are made, then they go back into these mouse models. We compare the efficacy of these new agonists that we've made with known agonists that we already know about that are beta-1 selective, and onto these what we call phase 0 human trials using organoid models, and they are patient-derived. So that gives us a lot of information actually about how well they can target this protein. For me, I have some specific questions that I wanted to address that I think are very interesting. So first, I took this molecule. It's just a bunch of letters and numbers, but I'll show you right here. It's PF06409577. PF because it was actually made by Pfizer, the pharmaceutical company, if you guys know that company. It is a beta-1 selective agonist, and it's much, much more selective for beta-1 over beta-2. Now, there are many beta-1 selective agonists, but I found this one interesting because it did make it to phase 3 clinical trials for diabetes, not for inflammatory bowel disease, because targeting AMPK, like I said, is also useful for many other diseases. And so the numbers showed here, using the EC50 as 7 nanomolar, a very, very, very small concentration for beta-1 efficacy, and beta-2 being quite large. And also the binding constant, KD, of 9 nanomolar. So that leads me to my second question, which is interesting for me. Does binding relate proportionally to activity? Because you see the EC50 is activity, and the binding is KD, but is that actually always correlated? Is the binding always correlated to the activity, or is there variation? So, sorry about that. My specific aim is to design, synthesize, and then test in vitro these novel beta-1 selective activators. And it will be guided by in silico docking studies. Give me a second. Sorry about that. Okay, so what I'll be doing is, like I said, with the aim is to design, synthesize, and test, and it will be based off of in silico docking. So what that means is using these co-crystal structures that have already been published and referencing our docking studies based off of the published co-crystal structure. Thankfully, lab member Ashish Shiv Kumar in the Yang lab has graciously helped me make most of the docking slides, and it's been helping showing me how to do these docking studies. And so this figure here is not a docking study, but it is a co-crystal structure of the molecule that Pfizer published. So we see here this molecule of interest in the binding pocket, and it's right in between the beta 1 and the alpha 1 subunit. And in black, I have highlighted the protein residues that it's known to interact with. And in red are all these other protein residues that are in a pocket around it. And so these are potential protein residues that we can reach out and grab and get more interaction and try to make it even more potent and more beta 1 selective. So in order to try to make it more beta 1 selective, we must then look at also beta 2 molecules and compare them in their selectivity. So some of these known molecules we looked at, and this is just a couple out of actually over 100 that Ashish looked at and analyzed. And so just to highlight a couple key features is this nitrogen here. Only being in the beta 2 selective molecules, we think this might be a nitrogen anchor or interaction with the beta 2 subunit. But we also see it here in the non-selective molecule. And we hypothesize that maybe this big bulky group might be pushing it out of the key zone for this nitrogen to interact with that beta 2 subunit. We also see the carboxylic acid group here for beta 1 selective molecules. So that was just a couple of many observations that we made. So we created a hypothesis based around the indole group. So this indole skeleton, I hypothesized different groups for its selectivity. And based off of co-crystal structures and the published co-crystal structures, we have known information about this third group here. Has a Lewis base site for lysine 29. This beta 2 selective region we hypothesize is the nitrogen site for this group right here. And then for most of these groups, as you can see here, they have a bulky hydrophobic group, which we just called the long arm. I think I don't have time to get through that, so I'll just go through these slides. So using docking, now we're into docking, we can compare the co-crystal structures. This figure right here is the molecule in the binding pocket. We know that this is the truth, and this is the docking, which is a simulation which is not always accurate, but it does predict the binding affinity. And so Ashish proposed that we add this ether chain, so oxygen and then extra carboxylic acid out here. And you can see that it's reaching out to this arginine 83. So that is an additional protein residue that we're trying to reach out to and see, will this make it more selective? As you can see here, the known molecule, the PF molecule here, does not interact with the arginine residue. So there was another molecule that we also predicted, and we basically wanted to test the maximum number of residue contacts. And so you can see there's a lot happening here. There is a possibility that this just, completely destroys the basically the protein pocket with too many interactions and whatnot. But we did want to test, can we reach out to as many protein residues as we can and see if that makes it a potent binder. So you can see here the known molecule residue contacts we know is 4. The predicted for compound 8, which was the last slide, is 4 to 6. This one is predicted to have 6 to 8, which is a lot. So lastly, I just wanted to go over the synthesis of how to make these molecules. So this is a published synthesis that I followed for making the known molecule. And first you start with 1,4-dibromobenzene. You basically just add the cyclobutanol group here, and you run a Suzuki reaction to make this boron group here. And then you start with the indole skeleton here, with the intention of using this bromine to then react with the boron for another Suzuki. So you add you isolate the 3 position here of the indole, and once you combine these 2 molecules using another Suzuki reaction, you get compound 4. And so you're very close to the final product. You just need to hydrolyze it with sodium hydroxide. And so then you get the PF molecule. So it's pretty straightforward. Not too many steps. And so using this kind of chemistry, I just wanted to derivatize this molecule with the same reactions. But it turns out that it was a lot harder than I thought. So this was my original proposal. And actually, the key reaction failed on many attempts. And so I was not able to get compound 6 here, which was like the main key factor. The main derivative is this oxygen chain right here. And so using a lot of different other reactions, I finally came to this reaction just last year before winter break, using this key intermediate with chlorine. And I was able to get this compound here. And so basically now that I have this compound, it is a low yielding reaction. I don't have much of it, but using this compound, I can go ahead and use a lot of the same reactions that I showed for the known molecule, combine it with the compound 2 that has the boron, make this molecule, and then hydrolyze it to the carboxylic acids. So that would basically be also the same thing for all these reactions. I don't want to go through all of them, but this is for the super binder to make the one that has the maximum number of residue interactions. Just a lot of steps, and of course, this key step is the most important to get to this compound. So now I will be using the previous reaction I showed you that did work to make this compound, and then go ahead with these other reactions that I have predicted and have high confidence that they will work. Okay, so lastly, like I shared, I will be doing in vitro testing. So it'll be an activity assay, and it will be known as the ADP glow kinase assay. And so what you do is you start with this, the substrate, ATP. And so for this, this assay, it uses a substrate called SAM. And the substrate takes the phosphorus from the ATP. So there's remaining ATP, and then there's ADP. So that remaining ATP, fortunately, we just try to deplete that and make that go away. The ADP then, you add the kinase. So for us, that kinase would be AMPK. That AMPK then converts the ADP into the ATP. And how well it's converting is that activity. So if it's converting a ton, its activity will be high. This newly synthesized ATP is then detected by the luciferase. So we add that into the assay. Luciferase is just a protein that shows a lot of light when it detects ATP. And then there you have basically an activity assay. It's directly proportional to the light, the absorption that you read using a plate reader. So that will be the in vitro testing that I'll be using to test the AMPK. And yeah, Ashish will also be doing that, probably most of the AMPK assay as well. Okay, so that's all I have the summary is basically to design, synthesize, and test these activators. We have 2 activators I showed you, and many more that we're still designing and trying to work on. And so I made the known molecule, and I started the synthesis of some of the derivatives, and then basically the next steps that I have are to finish these 2 molecule derivatives, to characterize them and test the binding and the activity, and then to continue the in silico docking experiments based off of the data I get. So it will be really helpful, kind of like a cycle of seeing that data that I get, and then going back to the computer and simulating and see where the inaccuracies and where are the validations. With that, that is my research. I'm really thankful that I was able to share with all of you. And I just want to acknowledge my co-mentors for Ghosh, especially Professor Amaro, one of my committee members, and the Ying Lab, and especially Ashish for helping me on this project, the Ghosh Lab, and the postdocs in that lab that have been helping me as well, and of course the UCSD Interfaces Graduate Program, and the NIDIB for the funding that I've received. So thank you, guys.
Thank you, Joseph. That was a great presentation. Any questions for Joseph?
I have one for Joseph. Yeah. Hey, Joseph, that was super cool, man. Thanks. I had a couple questions. They're more just like how you're planning to do things. That's super cool that you're synthesizing the molecules. And I guess I have 2 questions. So one is, where are you going to get the AMPK? Are you going to crystallize that from, stocks your lab already figured out how to purify? And I guess it's active, even though it has 3 components. I guess it's active just as the beta component for your kinase assays. It's another part, I guess, just the beta part can be measured without its other 2 components. And then I was just curious about the logistics of taking a molecule that was patented by somebody and modifying it. I mean, it's totally cool, I guess, to just add groups. There's nothing tricky logistically with that, since Pfizer obviously owns that patent. So thanks, man. Just curious.
Oh yeah, those are all great questions. Thank you. For the protein itself, my lab member Ashish has already actually purified the protein itself, and we are testing for beta-1 selectivity, but we'll be using the whole protein. So we'll just be using alpha-1, beta-1, gamma-1. You're right that it does have to be activated, actually. So Ashish was also able to purify one of its activators. It has 2, or we should call it phosphorylator, maybe is more specific term. Be able to make it even to do stuff, and that would be calcium, calcium modifying protein. I forget exactly. Sorry, but yeah, it's a calcium modifying protein that phosphorylates the AMPK. And then, great question about the patent. We dove into the Pfizer patents on this molecule, and we're very pleased to see that that 2 position. So if I can go back wherever I have the molecule, that 2 position. So with nitrogen being 1, the 2 position here is actually not patented. So yeah, that was what our assumption is. That's what we're going off of. Yeah, I guess. Yeah, I guess, does that answer your question?
Yeah, that's totally good. I don't know anything about having a drug patented, so I was curious. Yeah, if this is like pretty common in the field, basically, like, yeah, you go look for a group that hasn't been patented and go for it, and I guess that makes sense that's how it would go. But yeah, I guess you have to synthesize it all the way. You can't just buy Pfizer's drug or get some thing and modify it. I guess you're, to build your molecule, you have to like build it differently to get to where their new one. I guess that was interesting too. But yeah, thanks.
Yeah, of course.
Hi, Joseph. I wanted to ask a question about the Boolean gene network stuff.
I'll try my best.
So it's just like in the decision-making step. So at the very beginning on that, like, if you go a couple slides forward, actually, where it's like decision tree kind of thing. So did you guys choose PRK A beta 1, and you also mentioned PPAR because there's like some mechanistic reason, or because they had already had drugs associated with them?
Yeah, great question. For a lot of reasons, actually. So in cluster 2 is maybe, I think, 13 or 14 proteins that all kind of were, druggable targets. And yeah, I didn't go into the very specifics about that, but there's a lot behind just the word, druggable protein target.
Mhm.
Do they have known molecules that we can base our studies off of? Are they, molecules that are, or proteins that are easily purified, have papers published behind them, stuff like that. So, actually a lot of criteria, I think maybe it was 9 or 10, that was published in Pradipta Ghosh's paper. And so we just found that PRKAB1 was the best in terms of all the criteria. And we went forward with that, but there was maybe at least 13 proteins that fit that cluster in terms of a protein that could be targeted to stop the disease state from happening.
Okay, cool. Thanks so much.
Thanks, Joseph. So next up we have Nate Linden, who's going to talk about his Monte Carlo and Bayesian modeling.
Cool. Can everyone see my screen and my mouse?
Yep.
Yeah.
Okay, perfect. So I'm Nate. I'm a second-year PhD student in the mechanical engineering department. I'm co-advised by Professor Ranghamani and Professor Kramer, and I'll be talking about my ongoing work in Bayesian parameter estimation from sparse and noisy measurement data in systems biology. So I want to make sure everyone's on the same page as me about what I mean by systems biology 1, the types of models I'll be talking about, and what parameter estimation is before I get into the details of the Bayesian estimation algorithm. And I know this is a total change of pace from the last talk, so feel free to interrupt me at any time if you have any questions. I can't see the chat, so just yell out if there's any questions. So typically in systems biology, and the systems biologist will choose some system that they want to work with. So this is the schematic of a cell signaling system. And the goal is to derive some mathematical model. So in the case of my work, it's a system of ordinary differential equations that describe the time evolution of the concentrations of the relevant species, biochemical species within the system of interest. So the steps to building a model usually begin with determining the network structure. So what is our system and what are the relevant biochemical species? Then you use biochemical theory to derive a model structure. So we say we use mass action kinetics or Michaelis-Menten kinetics to come up with an equation that tells us how the concentration of these biochemical species evolve over time. And then lastly, there's some manual tuning or calibration steps to make the output of the model match the experiments. And historically, this is done by doing parameter sweeps or manually tuning the parameters until you get the desired or expected output. So my goal is to go from manual tuning or manual calibration steps to learning the model or the parameters directly from data. So this is kind of an idealistic data set. That is my end goal, to be able to work with like spatially resolved fluorescent imaging data. But there's a couple questions we can answer in this data-informed or data-driven modeling paradigm. So what are the governing equations of my system? What are the appropriate parameters given some model formulation? How do uncertainties in the data the model or the parameters affect my predictions I make. So uncertainties would be noise, or maybe I don't exactly know the model structure or the parameter values. I only know them within some error bound. And then lastly, can I develop an equivalent model with fewer degrees of freedom? And each of these questions falls into some subcategory of computational science. So learning the equations is model discovery, parameter estimation is choosing parameter values, uncertainty quantification is understanding uncertainty, and reduced order modeling is reducing the number of degrees of freedom you need in your model. So today I'll focus on parameter estimation and uncertainty quantification. So diving into the parameter estimation problem a little deeper, you generally start with a model. This is a schematic of some system, and equivalently, there's a set of differential equations that govern how each of the concentrations of these molecules changes over time. And those differential equations have parameters, so that may be rate constants or the binding affinities that govern those equations. So that would be like the KD value that Joseph talked about. And then we have data, so we do some experiments where we measure a subset of some of the molecules in our system of interest experimentally. And then our goal is to fit the model to this data. So we want to make these solid lines match the points as best we can. But there's some major challenges associated with biological data and biological systems that make parameter estimation particularly challenging in systems biology. So first is that noise is unavoidable. So if you look, the error bars in this data can be quite wide, and that makes parameter estimation more difficult. The data are sparse, so we have very few time points in the data, and we also have incomplete data. So we can only measure a subset of the biochemical species in our network. So in this case, I'm only showing time series from 2 out of the however many there are in this network. And lastly, measurements are often made indirectly through a quote unquote nonlinear sampling process. So if you look, the y-axis in these data are not direct concentration of molecules. There's some fold change or some, it's not directly the concentration value. So that adds a measurement process into our, that we must account for in our models. So next I want to talk about, or I want to set up the mathematical framework for parameter estimate that I'm going to use for parameter estimation and just for thinking about systems biology models as state-based models. So like I said, our goal is to come up with some equation that governs the dynamics. So X is the collection, it's a vector, it's the collection of concentrations of all the biochemical species in our system. F is the nonlinear model, so that's our differential equations. It can depend on the state, the time, and then this theta F, which is the collection of model parameters. And our goal is to learn or to estimate this theta F vector. And then we make noisy measurements. So Y is our collection of measurements. H is our potentially nonlinear measurement operator. And we assume that the noise is independently identically distributed mean zero Gaussian noise with a covariance vector gamma. And then since we said that we may have fewer measurements than we have states, M can be much smaller than the number of states D. So the typical or the most intuitive way to look at parameter estimation is to say, I want to make my predicted dynamics, so that would be Y hat, look as close to the measurements as possible. And when you ask that question, you end up formulating the problem as an optimization problem, where you say, I want to minimize the distance between my measurements and my predicted measurements in a least square sense. And then you choose the set of parameters that minimizes that distance. So in this point of view, there's a single optimal parameter set. that we don't know when we start, but you can use an optimization algorithm to find that single set, which may or may not be true in biology. There might be a single set that governs our system, a single set of rate constants or binding coefficients that governs the system. But typically in this setting, we don't get a good sense of the uncertainty in this measurement. So you can estimate error bounds with assumptions of linearity, but you don't get a true understanding of the uncertainty in theta. So alternatively, we can take a Bayesian approach to parameter estimation, where we will want to learn a probability distribution for our parameters. So that's the probability of the parameters given or conditioned on the data that we observe, which is known as the posterior distribution. And in this point of view, the parameters are random variables. They can exist over some entire parameter space, and they're governed by some probability distribution. So our goal is to find and characterize this posterior distribution given the data we have available. And we can find this using Bayes' rule. So Bayes' rule says that our posterior, which is the probability of the parameters given the data, is equal to this product of distributions on the right. So we have the prior, which is our assumed probability distribution. So the most intuitive prior is just to say we know our parameter can be within a lower bound and an upper bound. So you'd use a uniform distribution for that. And then that's multiplied by the likelihood function, which measures the probability of the data given the parameters. So essentially, you estimate the state and you compute a similar norm to what I showed in the optimization-based case. And then you have this denominator, the probability of the data, which is often unknown when we set out, and we usually just ignore it and replace a quality for proportionality. So this should be a proportionality symbol. So our goal is to formulate this right-hand side so we can characterize the posterior distribution. And given the formulation of where we just have noisy measurements, you can write an expression for the likelihood function. As I said, we know the prior when we start, because that's what we assume we know about the parameters. So we come up with this formulation, the likelihood, and I'm not going to go into the details of what all of these means. I'm happy to share more information with anyone who's curious afterwards. But this is the expression for the likelihood function for measurements with Gaussian noise. So we could construct the posterior distribution in this case. One thing to note is that this is not an explicit formula for the posterior. So unlike if we just assumed we had a Gaussian distribution, we could come up with an explicit formula and just evaluate that at every point in parameter space. The only way to resolve the posterior is to take samples of theta, plug it in to this expression, and get a value for the posterior. So what we end up having to do is we sample from the parameter space and study the statistics of those samples. And we do this using Markov chain Monte Carlo sampling, which for the sake of time, I will avoid the details of today. There's one major caveat with what I've talked about is that we assume the differential equations, the model is perfectly correct. We don't leave any room for the model to be incorrect in the formulation where we just assume we have noise or measurements. So in order to account for this potential model uncertainty, we're going to introduce a process noise term. So everything here looks the same as the slide I showed before, except we now have this C noise vector added to the dynamics. So this is the process noise. Again, we assume it's Gaussian because that allows us to use very nice Kalman filtering methods. But in reality, the process might not be Gaussian, but that's one of the things we assume. And then our goal is still to learn theta f from the data. So now we want to ask, what is adding this process noise change to the coming up with an expression for the likelihood function? And it turns out it changes a lot because now both the state X and the measurement Y are uncertain, or they're random variables. So we have to then marginalize out our uncertainty in the state, and we end up with this very high dimensional integral, which there's not a clear way to solve this integral by hand or even numerically. So in order to approximate this integral, we'll use a common filter or a nonlinear variant of the common filter known as the unscented common filter. But in order to avoid getting into too much math, I won't talk about that today. One thing I want to point out is that this method of adding in process noise has been shown in the past to improve parameter estimation from noisy and sparse input data. So essentially, this is the overview of what the unscented common filter does. There's 3 steps. You solve this recursively over your data. So meaning you have a for loop and you loop over all of your data. You evaluate these probabilities and you get the likelihood at each time step, and you multiply them together to get your overall marginal likelihood. And I'm not going to go into the details because it gets very notation heavy very fast. So we now have an algorithm to construct a posterior distribution for our parameters given a data set that accounts for both measurement uncertainty and potential model uncertainty. So we defined a way to evaluate this posterior for a given instance of theta. And one thing I want to point out is in order to evaluate the posterior once, you have to loop over all your data and evaluate the model at each, like over the entire time domain that you're interested in. So this is many computations for one posterior evaluation. Like I said, in practice, we solve this recursion using a common filter, and which could be approximated for nonlinear models, which are the interesting models in systems biology, using the unscented common filter. This does not give us an analytical expression for the posterior, so we have no explicit equation, so we have to sample again from this distribution using MCMC sampling. So I think the next important question is to ask, how does this parameter estimation, which is the UKF MCMC algorithm, how does that fit into a model development pipeline? So like I said before, the typical process was take a system, formulate your model, and then manually tune parameters to make the predictions match the data. Now we get this 6-step process. So again, you formulate your model. I assume we're able to come up with a model formulation based on our first principles understanding of the biochemistry and of the biology in the system. You do some experiments or you collect experimental data. Then we do what's known as a priori parameter analysis. So I'm not going to talk about identifiability or sensitivity analysis, but essentially that is, can we actually learn the parameters from the data that we have available, or do we need to fix them at something and ignore them? Next, we gather all relevant info on the parameters in our model to formulate the prior. So in Bayesian estimation, if your prior is really good or it's close to the true distribution for the parameters, you're going to do a better job, meaning you're going to have less uncertainty in your estimates. We then run the UKF MCMC algorithm, and we can analyze the posterior distributions for the parameters that we get. So to illustrate this process, I'm going to work with this very simple linear 2-compartment model. So this essentially describes transport between 2 isolated compartments for a single chemical species. There's transport rates between the 2 compartments. Out of compartment 1, there's some input in the compartment 1. And we assume that we can only measure the concentration in compartment 1. So from this, we want to learn these 3 transport rates, K12, K21, and K1 external, and the coupling rate for the input into X1. So I ask two questions about the system. One, what happens as I increase the measurement noise? So these points are the measurements I'm making. What happens if they get further away, like they spread out more around the true trajectory, which is the solid line? And then also what happens if I decrease or increase the number of samples in time that I have? So first, we're gonna look at what happens if we increase the measurement noise. So I'm gonna take a second to explain the plots we're looking at. So this is the trajectory. The true trajectory is the solid lines. And essentially the range of possible predictions, or 95% of our predictions, lie within these shaded regions. So that gives us a sense of our uncertainty and the predicted dynamics. So when the narrower these bands are, the more certain we are about what's going on, and the wider they are indicates more uncertainty. So we see that essentially as we increase noise, the noise increases going to the right and down, our uncertainty interval grows to the point where things completely break down at 10% measurement noise, indicating that the noisier the data, we can't predict where the trajectory is going to be as well. And then this also can be seen if we look at the posterior distributions for each parameter. So there's each row is a different parameter. The posterior distribution is the green distribution. We see that as we increase noise going to the right, the distribution gets wider for all the parameters, meaning there's more uncertainty in our estimate. The next question is, what if we increase the number of time points, or going this way, increase sparsity in our data? So we took 10 time points, 20, and 40 time points. And you see that, again, as you increase the number of samples, our uncertainty shrinks. So we're able to make a better estimate the more the data we have. So today, I talked about a Bayesian parameter estimation algorithm that, with the addition of a process noise term, can improve parameter estimation from sparse and noisy data, but it requires some recursive algorithm to evaluate the posterior. This algorithm allows us to capture the uncertainty due to measurement noise in data sparsity. And my future work will focus on using this algorithm to estimate parameters from experimental data, and then establishing it within a parameter estimation and uncertainty quantification pipeline for more generic systems biology models. So thank you for listening. Does anyone have any questions?
Nice. Thanks for a very nice presentation, Nate. We have time for some questions. So how many parameters have you tried to estimate?
So right now, I'm focusing on relatively small parameter spaces, so on the order of 10 to 15. As you get more than that, standard MCMC techniques start to break down.
Mm-hmm.
So I'd have to switch to variational approaches.
Any other questions for Nate? Seems like your tools could be valuable to a lot of students in the program, including some of mine. So
yeah, that's the hope, that it's accessible.
Yeah. Okay, well, if there are no more questions, thank you, Nate. Thank you, Joseph, for great talks, and thank you to everyone for coming along. And we will see you next time.
Thank you, everyone.
Anybody want to vote on funniest hat? I guess they all left. I'll take a poll of the group. Thanks, Nate. You did a great job.
Thank you. And then I wanted to remind you about the release that you told me to remind you about.
You're wonderful. Thank you so much. I appreciate it. And I'll do that in just a moment.
So thank you.
Thank you. Bye-bye.
Bye-bye.